Well, good morning. It's great to be back at OIS, and I want to thank the organizers for giving this opportunity to come and talk today. Today, I'm going to share our ongoing plans to develop a pipeline of long-acting retinal medicines with the goal of reliable treatment once every 6 months with a treat and release protocol, and how we believe we're setting this up so that we can provide that real reliability of dosing for all patients, regardless of the patient variability we know exists. So we've heard a few different strategies for extending the durability of retinal medicines this morning, and so I want to also start by pointing out what makes Valitor different. So our compositions have all been designed so that they can be administered with a standard intravitreal injection protocol. We use 30-gauge needles and a 50 microliter injection volume. The difference in our compositions is that they're intrinsically designed to eliminate from the eye slowly and at a defined elimination rate that gives us the clinicians confidence that they can treat their patients and release them for 6 months and know they don't need to have them coming back for screening visits, and they can get their dose on schedule 6 months later. Our compositions are all dispersive and completely transparent. There's no risk of optical interference. And we've designed our compositions with safety in mind from the start, using only endogenous materials. And that's now bearing out in our non-human primate tox studies that are ongoing. Our chemistry for generating these compositions is plug and play, which enables us to work with internally derived or partnered assets to rapidly establish a pipeline of medicines. And to that end, we're currently working on 3 different product programs. The lead is our internal asset, VLTR-559, which is a long-acting anti-VEGF designed for once every 6-month administration. It is currently ready to go into GMP manufacturing and into clinical trials. We, in parallel, are working with 2 industry collaborators who have APIs that need durability for differentiation in their space. We've demonstrated that with our platform, we can generate highly potent conjugates, and we're currently working on license-enabling milestones. We don't plan to stop there. We're always looking for new APIs that need durability. We can then follow the established development pathways in order to deliver long-acting versions of those assets to the patient populations who will need them. So I want to spend the next few minutes talking about the preclinical data we have, giving us confidence in the reliable re-administration schedule that we believe we'll have for our lead product, VLTR-559. This is highly representative of all the other products in our pipeline as well. As I said, it's a long-acting biologic anti-VEGF. It is designed to leverage that gold standard biologic anti-VEGF mechanism that we know works well for most patients. What's needed in this space is longer acting drugs, and VLTR-559 is designed to meet that need. The anti-VEGF biologic we use is a single domain antibody that gives us the smallest, most stable unit of a biologic to give us that anti-VEGF specificity. And we conjugate multiple copies of those along a backbone of hyaluronic acid. We like hyaluronic acid because it is biocompatible and endogenous, and it really maximizes the interaction that we can have with that back of the eye vitreous matrix. By putting multiple copies of the drug along the backbone, we can avoid any biopolymer interference and, in fact, increase the potency of the antibodies on a molar basis by creating these multivalent interactions between the drug and the target. And we know the linear nature of the hyaluronic acid enables it to penetrate deeply into tissues like the retina. We've studied this composition extensively in preclinical models. This is the result of a laser CNV model. In this experiment, we used humanized mice that express human VEGF, so we were able to do a direct head-to-head comparison, showing that VLTR-559 has equivalent ability to inhibit that VEGF-induced neovascular response to Eylea. So from a potency basis, these molecules are exactly as potent as what's currently available on the market. Where we differentiate ourselves is on the basis of our durability. So this is a PK study in rabbits showing a very consistent elimination rate that's also consistent with all of our previous PK studies on these compositions, with a half-life on the order of 12 to 15 days. That's in comparison to something like Eylea that has a half-life of about 4 days. So we can extend the retention of the drug right on target around 3-4-fold relative to existing drugs. And we often get asked, well, how do you know that the drug is still active while it's in the eye? We're reporting today for the first time we've actually extracted the VLTR-559 out of these PK samples, and we're able to now demonstrate that at the end of the study they have the same potency as when we put them in. So we've got rock solid stability of the drug, which was built in by design, and that's now bearing out. So we can project how this will impact patients using clinical efficacy models. This tracks or predicts the concentration of the drug in human eyes over time after administration. And you can see, again, we've got a very predictable slow elimination rate that is keeping the drug at higher concentrations longer than the currently available alternatives. And we can predict when these drugs need to be redosed based on all the previous anti-VEGF registration studies. And based on this is what's giving us the confidence that we'll be able to dose the patients reliably. 90% or more should be getting to 6 months without needing readministration. Then they can come back in, have their visit, and get redosed. So we're excited about what we're able to do with VLTR-559 for wet AMD patients. I want to spend the last minute talking about the platform that enables all of our pipeline. We call it multivalent polymer conjugation, and it enables us to take a variety of different drug formats. We've validated that it works with all of these, and especially large molecules, to enable us to use our interchangeable chemistry, conjugate it to the long-chain hyaluronic acid, and in doing so, we can bring all the benefits of VLTR-559 to new APIs and indications. So our goal is to continue building out this pipeline of long-acting retinal medicines. Again, we're excited to be here today because we're looking for investors who see the value in this pipeline and would like to help join us in order to move this forward into clinical development. We're also always looking for new business development partners who have APIs that need durability. We'd love to see how we may be able to help you. If any of this sounds interesting, please come find one of us during one of the networking breaks. We'd love to start a conversation. Thank you.